Well, welcome friends of TMAG to this. This is our first virtual floor talk for 2020. In fact, the first one ever, you might realise. So today we're going to be talking about Tasmanian seashells, and I'd like to welcome Dr. Simon Grove from the Tasmanian Museum and Art Gallery. So Simon is the Senior Curator of Invertebrate Zoology. And today we've got a PowerPoint slideshow that we're going to chat away around as we work our way through it. And Simon will lead us through that process. So I'll start us off. So welcome, Simon, and thank you very much for joining us.
Thank you for the invitation, Janine.
Okay, so the first thing is, Simon, I gather that you had a very good childhood shell collection. So can you tell us a bit about the first shells you collected and this childhood museum?
Yes, I was pretty besotted with shells from quite a young age. I think probably the first shell I remember collecting was a cowrie or a cowrie relative on a beach in Cornwall in England, where I grew up in England. And I was just captivated. I had a Ladybird book of the seashore or something like that it was called, with beautiful, beautiful paintings in it, which really just were very evocative for me. I just loved beach combing and picking things up off the beach and finding out what they were. So that was the start of it, really. When I was about 8 or so, my granny gave me and her other grandchildren a little bag of Polynesian seashells, they were called, and they just seemed so exotic and so wonderful. That sort of triggered me into the shell collecting habit. So by the time I was 9, I think, that's when that photo on the left was taken of me with my own little shell museum in my bedroom back in England. And we stuck the sign that's there up at the end of the road in case any passing shell enthusiasts wanted to come in and have a look at my shell museum. I can't say that many did, but the offer was always there.
That's great. And how did you get to the Tasmanian Museum and Art Gallery from such beginnings?
Well, shells were a big part of my childhood, but as that little handmade seashell book in the middle and the right-hand parts of that screen demonstrate. But by the time I got to my teens, I'd moved on to birds and then butterflies and other insects, and then more natural history more broadly. I did study marine biology at uni, but after I'd finished studying, I got work in terrestrial nature conservation, pretty much in invertebrates with the National Trust in the UK. And it wasn't until I studied forestry for a master's that I started thinking about travelling overseas from the UK. I ended up working in Uganda and then Indonesia, and that's when I got offered a scholarship to do a PhD in Australia, James Cook Uni. So I moved there, and out of that was on beetles as it happens. Out of that I got my job down in Tasmania with forestry as a conservation biologist, and that's when my interest in seashells was really rekindled, because Tasmania's beaches were just a joy to walk along, nice and peaceful and quiet, and so many seashells on the beaches, on many of the beaches, that it was just crying for someone to come along and figure out what they all were. So there were a couple of guidebooks at the time written by Margaret Richmond, but they were hard to get hold of, and they were out of print. So before long, before I knew it, I was building up probably the best collection outside the museum for the Tasmanian seashells. And that's how I came to write the book on seashells, which we'll talk about later. And it's probably on the strength of that when the job came up at the Tasmanian Museum, I was offered the position as senior curator. That's how it all came to pass.
That's a wonderful story. Thank you.
I'm very privileged.
Could you tell us a little bit about the history of shell collecting in Tasmania for scientific purposes? Who were some of the leading lights, and how far back does it go?
Well, it goes back to really the early days of the Royal Society of Tasmania, and this character here called Julian Tennyson Woods, who was a Catholic priest who seemed to have a bit of a fiery temperament, I think, because he had been, at one stage, he'd been over in Victoria, Penola, at the same place as Mary MacKillop, but I think he fell out with Mary MacKillop or someone of that ilk, and was sent to Tasmania. I think because he didn't get on with people over there. So anyway, I'm not quite sure what his pastoral duties were here, but he took to geology and to shell collecting, and he published large numbers of papers, particularly in the Royal Society Journal, and he was responsible for describing most of the first of the Tasmanian shells, other than the early explorers who had taken specimens home to Europe. He was the first sort of indigenous, I can't use that word, he was the first person living and residing in Tasmania who ended up describing Tasmania's seashells. And he eventually produced a list which listed 523 species, including land snails, but mostly seashells.
Yes, yes, that's amazing.
And yeah, that was, I'd say he was a bit of a funny character. He named a lot of his seashells after Catholic saints. So there's things like Theresii and Vincentii and Aloysius. He was quite keen on his Catholic saints. Anyway, his research and his publications stood the test of time for the next 30 or 40 years. And then it was passed, the baton was passed to a Hobart-based businessman called William Lewis May. He was a trader in apples and I think boots. I think he exported apples and imported footwear. But his passion was marine mollusks. And he had the wherewithal to actually go and do some of this, do surveying on his own by chartering a steamboat. He worked with the Tasmanian Field Naturalists Club as well, and some of the expeditions were joint ones with the field naturalists. And he teamed up with a malacologist, that's a mollusk expert at the Australian Museum, called Charles Headley. And as a result of their expeditions, they went dredging off the Tasman Peninsula and off Maria Island and a few other places. They really bumped up the list of known Tasmanian marine mollusks, and they produced an annotated checklist, which pretty much doubled what Tennyson Woods had known. This is a page from that checklist, and I think I've just highlighted a few there that all come from what was then called Thuan Bay, which is now called Wineglass Bay. I think that's the same place.
Oh,
so dredging in 50 fathoms, I can see there, 40 fathoms. A fathom is 6 feet, so 2 meters, so about 80 meters depth. And so these are shells which would never wash up on Tasmanian beaches. They were unknown to science at that point, but they described a lot of them in their publications.
That's fabulous. So thinking about collecting now on Tasmanian beaches, how do you go about collecting scientific specimens, and what sort of diversity is there out there on our beaches? Yeah,
well, you could expect, if you really got into it, you could expect to find perhaps 400 species on Tasmanian beaches around the state. You wouldn't find 401 beach, you'd be very lucky. But the best places to look are where there's a mixture of rocks and sand, and where the tides or the currents are just right to bring in the dead shells, because what we're looking at is the dead shells of animals that are living either on the shore or just in the shallow subtidal area. When they die, the shells roll around in the waves, and if you're lucky, they get washed up. So finding good beachcombing spots is half the art, and then the other half, which is perhaps more of the science, is learning how to identify the things and to recognize what you've got. That also is only part of it, because if you want it to be a good scientific collection, you need to document it as well. So most people will stop at saying, well, I love cowries, I'm going to have a jar of cowries on my mantelpiece or something like that. That's great, but if I walked into someone's house and saw a jar of cowries and I spotted a rare species amongst those cowries, I'd want to say, oh, where did that one come from? Who found that? And when was it collected? So if you haven't got that documented, the scientific worth of that collection is not as high as it could be. So if you want it to be a scientific collection, you really need to record where and when and by whom everything was collected, which of course means labelling and keeping different lots from different collecting dates separate from others. Actually knowing what species it is isn't as important, because that can always be done later. But if you lose that initial information about where, when, who and how, then the sort of long-term scientific value is diminished. But there's nothing to stop you. I mean, there are things to stop you, but there's no harm in collecting just for personal pleasure. But if you want something to have a bit more scientific value in the long term, then there's an extra step you need to go. So this chart shows how many species live intertidally on the left, and there's relatively few that do so, only about 100. But those species probably found nowhere else. So intertidal habitats is very different from anything that's permanently submerged. And we might think of those as the common species, but probably in terms of relative abundance, just because the intertidal area is such a thin strip, those are probably the rarer ones, whereas the things which we rarely find washed up on the beach because they live offshore are probably more widespread, got a wider geographical range because there's more of that sort of habitat around. So although the shape of this graph goes upwards from left to right, the relative rarity is probably the other way around in absolute terms.
Absolutely. So I guess the message for people is if they think they've found something particularly rare, they should bear in mind that idea about noting where they found it and under what sort of circumstances. That's
yes,
interesting.
That's right. And if you think you're going to keep this collection for your life, then make sure that someone else knows about it, so that if there comes a time when you need to pass the collection on, it's not chucked in the bin, because that happens as well.
Yes, yes, I'm sure. There's some lovely, lovely pictures of you collecting here.
Yeah, my first decade in Tasmania, which was the noughties basically, when my sons were much younger, we used to go out shell collecting quite a lot as a family. So these treasures, well, that's my son Ben, who's himself a bit of a treasure, but he's got treasures in his arms. He's got abalones and these beautiful umbilicated cowries, which are a real Tasmanian treasure. The range of the species goes all the way up to probably northern New South Wales, but at that latitude, they're only found in really deep waters, whereas down here, there are a few select places, which I'm not going to reveal, where you can find them in shallow water and even get washed up on the beach sometimes.
Fantastic. Beautiful.
This little chart on the left, can you see how I'm kneeling over, looking very intently at the sand?
Yes.
And the curve of my bottom. My wife Chris decided that she had to take a picture like that because it reflected the shape of the curve that I'd showed her for the cumulative number of species that you get through successive visits to a beach.
That's wonderful.
So that chart on the left is from Taroona Beach, and that's me on Taroona Beach. The image has been reversed for those who know the spot, just to make the shape the same. But yeah, basically, if you just go to a beach once or twice, you'll find some things, but you'll just be touching the tip of the iceberg. And if you really want to know what's there, you have to keep going back again and again and again. And mostly you'll be recording the same things again and again, but each time you go, you'll be recording something different as well. And that shows that it took me 75 or 70 visits to reach close to 350 seashell species on Taroona Beach, which is quite an astounding number of species for that little beach there. And most of those species were on that little stretch of beach, which you can see on the right there, and that very fine, what we call shell grit, which gets washed up in certain tides. That's where you find most of the species, because most of them are teeny tiny little things, not the big whelks and winkles and things like that, but much, much smaller.
That's wonderful. I think it's really lovely to see an image of a beach that we probably all know very well and to see what's actually there. That's great. Okay.
Yeah, so there's Ben again. A good few years ago now. He probably wouldn't want to see that picture these days, but when you're that small, your eyes are already close to the subject. And he had amazing eyesight at picking up these tiny little micromollusks without glasses or without a microscope or anything. I certainly can't focus closer than about a metre these days, but if you scoop up a handful of that shell grit and sort it, dry it out preferably, and sort it under a microscope or a hand lens, you can, with the forceps, you can pick out the tiny little micromollusks, which is what that little sample in the bottom left of the slide consists of. Those are little micromollusks pulled out of a handful of shell grit. And the main chart there shows that there's a whole size range of species that you might find. These are just from Taroona, going up to about 300 millimetres, 30 centimetres for the big shells, but on the left-hand end of the axis, down to 1 millimetre. And anything, there's no strict cut-off between what's a micromollusk and a macromollusk, but generally, if it's below about a centimetre, it could be called a micromollusk. Bearing in mind that some tiny shells are babies, are the ones that get big, but most of them aren't. Most of them are, that's as big as they get. They're adults and they're micromollusks.
Wonderful. Okay, I guess we're not sure whether it's okay to ask a scientist whether you have some personal favourites. But I've taken the liberty of putting a couple of mine into the slideshow here. So the paper nautilus, which I remember finding a beautiful specimen washed up on South Bruny Island that I was so amazed something so fragile could have probably come from Bass Strait, I imagine, floating its way through the water and arriving intact such a long way away. So do you have a particular favourite, Simon?
Well, I approve of your choice. Just to clarify with the paper nautilus, it's a weird sort of shell. It's not a seashell in the traditional sense because it's actually made by a female octopus, a pelagic octopus that lives out in the ocean. So she would have been carried probably down the East Australian current and ended up in southeast Tasmania. And her last act would have been to make this beautiful brood chamber, which is what the nautilus shell is, for her young. And in this case, it would have been washed up, hopefully, once the nautilus had died and the young had been liberated. But that's not always the case. Sometimes you do find the octopus still living in the shell.
Right.
As to my favourite, I think it had to be one of the cowries, and probably that umbilicated cowrie, which I showed in the previous slide. I think cowries, there's something sensual about cowries, and the tropical ones are gorgeous and all well and good, but I just think it's wonderful to live in a part of the world where there are cowries and where we're still in the temperate zone, and yet there are these amazing cowries. We have at least 6 species that can be found on Tasmanian shores and a couple more undescribed ones living offshore. It's a real treat to be living somewhere where you can find these cowries. Those are my favourites.
Absolutely. The next slide's about shell collecting, because a lot of people will have this debate about whether you're actually allowed to collect shells in Tasmania. I know some states seem to have quite strict rules around it. So what is the situation, Simon?
Yeah, well, so far in Tasmania, the rules are less strict than in some interstate states. If you're not walking in a protected area like a national park, then the default position is that you're allowed to collect dead shells on the beach. As to live shells, there are bag limits for most species, so you have to look. It's worth looking at that website there, which tells you what the bag limits are. And there are a few species which you're not allowed to collect at all. So you're not allowed to prize limpets or elephant snails off the rocks, for instance. There are some species where the bag limits are unlimited. So pest species like the Pacific oyster, you can have as many of those as you want. But other species, there are limits. But that website, it's worth going to, it's worth pointing other people to as well, because quite often these days I do see people wandering along with big bags, collecting everything in their path. And I can't imagine it's even just for local consumption. I imagine it's for the market. And that's just not on. It's really denuding our shores. And these creatures, they're not just living in isolation, they're forming an integral part of the rocky shore ecosystem. And if they're removed, then everything goes out of whack.
That's an important point. Thank you. So if we move on to collecting in deeper waters now, so if you want to go out there to see what's living offshore, how do you go about that as a scientist?
Can I just go back to that slide? Yeah, I guess one thing I meant to say is that it's often argued that seashells provide homes for things like hermit crabs, so even picking up empty ones isn't a great idea. In Tasmania, generally, that's not the case for shells that get washed up on the beach, because we don't have, unlike tropical areas, we don't have intertidal hermit crabs. We don't have hermit crabs that wander around when the tide's low, wander around on the beach. You do get those further north, but in Tasmania, we don't. I think the only exception is the really large shells. They do often house the beautiful big orange-coloured hermit crab, and those shells probably are limited, a limited resource for the hermit crabs, so don't take too many of those. But the little ones, I don't think it matters. Other than that, the more you take, the fewer there are for other people. So
yes.
Nice for people to be able to see shells and maybe collect a few, but there's no need to go overboard.
No, and I often think as our tourism numbers increase, that if everybody collects a shell or two, then yes, the numbers that will be there will be much more limited.
That's right.
Okay, all right, so happy to keep moving?
Yep.
Okay, so we're heading offshore now to collect shells?
Yeah, well, I had an inquiry to the Seashells of Tasmania website, which I maintain, which we can talk about later, well, probably 15 years ago now, from Rob Little, who wanted a particular shell identified, and it sparked a friendship that meant that a couple of years later, once he had found all the shells he thought he could find on Tasmanian beaches, he wanted to find yet more. He'd seen those shells in the books by May and Tennyson Woods, and thought, I want to try and find those as well. They live offshore. I live in Port Arthur, and those shells were found off Cape Pillar, so let's go look. He had his own boat, the Rambler, 10.4 metre boat. So he and I used to go out dredging for these. We had a special permit from De Pipri, which homemade dredge, which I think is on the next slide. Here we go. And we got a permit to see what was down there now. And it's quite amazing what was down there. It's for a sort of thrown together dredge and just a cray winch on the dredge, on the boat, and a polypropylene rope to haul it in and out. We did amazingly well. We did have to go out quite a lot, and after a while I decided it wasn't for me. I was just getting seasick whenever I went out, so I left that to Rob. But over the course of, I think it was about 15 or 20 trips, voyages you might call them, multiple dredges per trip, we accumulated close to, well, it eventually got up to 600 species of mollusks. These are all from the waters off Port Arthur, between Cape Rowell and occasionally up into off Fortescue Bay, but primarily off Cape Pillar and Eagle Hawk Neck, that sort of area.
That's an amazing number of species.
Absolutely, in about 20 to 100 metres of water. 100 metres is about as deep as the dredge would go with that arrangement. The small boat, not very powerful engine, and not very powerful winch, and a polypropylene rope. It was hard to get samples from deeper than that.
That's amazing.
There's a big jump in that net, if you can go back one again, sorry. A big jump in the number of species found. That was after a few trips where we started deploying a little fine meshed net bag behind the main dredge. So with the main dredge, anything that was less than about 10 millimetres or 1 centimetre was getting caught up in the dredge and getting washed out through the back of the dredge. So we weren't getting the micromollusks. When we put that fine meshed net bag in, just a tiny little one, that's when we started getting a big jump, because we were collecting the micromollusks as well. So you can see the curve took a jump and then it continued to climb, and it was maybe just starting to tail off at the very end, but really there's probably plenty more down there.
That's amazing. Wow.
Yeah.
So just putting it in that perspective again of, between the intertidal to the offshore zone.
That's right. And a lot of these species, it's the first time they've been seen since William May had been out there 100 years previously. So it's nice to confirm their existence, but we also found a lot more species that he hadn't found.
A question that I had to ask, which was about whether any sea mollusks are threatened species, because we often think about plants and, plants and animals, land-based, terrestrial plants and animals as being threatened, but we don't often think about threatened species in the ocean.
Yeah, if we go back to the previous one, actually, I can answer that question with that. These were 35 of the species which are either new to science or new to Tasmania. In fact, we had 100 species in that category. I think these are the 35 which we think are new to science. And why hadn't they been found before? Well, because they're hard to find, but does that make them rare? And that's an interesting ecological question as well as a sort of conservation question. Just because something's hard to find, just because it's rare in samples, that doesn't necessarily mean that it's ecologically rare. But on the other hand, some things can be ecologically rare, making them vulnerable. But as often as not in the marine environment, we don't know, we don't have enough information. But what you tend to find in almost any sampling you do anywhere is that you'll find 1 or 2 really common species, and then you get quite a lot of species which are sort of intermediate abundance, and then you get a great long tail of species which are rare, very rare. And you have to think, well, why are they rare? And how can a species survive if it's so rare that you only find it once when you do, 100 samples? I think if you go on to the next slide or two, you can see those now. Yeah, so this is the species rank, what do they call them? Rank abundance curve for all those deep sea mollusks that we collected off Port Arthur. So the one on the left is the first number 1 rank. So that's the most abundant or most frequently found. That was found on 70 occasions. And then as we go down that curve, we get to the species which were only found once altogether. Strangely, or perversely perhaps, the species that was found most frequently was the only, as far as I know, or one of the few threatened marine mollusk species officially listed in Tasmania, which is Gunn's screw shell. If you go back to the previous slide, there's a picture of it there.
Okay.
That shell has been a bit, the picture's a bit distorted. It should be taller and thinner than that, but...
Okay. My photo, my preparation probably.
That's okay.
What types of things affect mollusks then? What are the threats coming from?
Okay, well, this species was listed as threatened because there was a perceived threat, which was the introduced New Zealand screw shell. That's been rapidly expanding around Tasmania. It probably got introduced with oyster spat, which was brought from New Zealand to the Derwent for on-growing, and presumably there were young screw shells in there as well. They can occur at really, really high densities, and the idea is that, well, it must be out competing this native species, and it may well be doing so. But that chart on the next chart demonstrates that it hasn't managed to do so yet, because the Gun screw shell was actually the most frequently found species.
Right.
So that's one threat. There would be things like dredging, as in commercial dredging and trawling, but things which grossly disturb the seafloor would definitely be a threat to some species, introduced species. But the big elephant in the room these days really is climate change, I would say. The waters of eastern Tasmania have warmed by a couple of degrees just in the last 50 years, and the trend's speeding up. So, I mean, that's quite exciting, because it means that we're getting tropical species suddenly appearing on Tasmanian shores. And it's amazing what turns up here now, which didn't even 20 years ago. But on the other hand, if you're a cold water adapted species, you've got nowhere to go if the waters are warming. You can't keep moving south because there's no land, there's no shore for you to occupy. So those are the species which are potentially threatened, in my view. But it's very hard to actually gather the data to show that there is an actual observable decline, when most of these species are so hard to find in the first place. And it's very hit and miss as to whether you find them. That's why I call it like looking for a needle in a haystack. It's very hit and miss if you find them. So an absence doesn't necessarily mean they're not there.
No.
Presence means they are there, but absence doesn't mean they're not there.
No. Okay.
I can just say something about these rare species, because I find this fascinating, that some species are naturally rare. You'd think that evolution or genetics would want every species to be really common, because then you're maximizing your gene pool, and that's what genes want to do. But not every species can be common. So the other strategy that species use is to specialize. And that means that as long as things stay the same, they can find a little specialized niche, and they can sort of fly under the radar of predators and parasites and things like that, and they can just keep ticking over in their own little specialized niche. And that's all well and good as long as things don't change. But when things do change, for instance, climate change, or someone comes along with a big scallop dredge or something like that, I would think that those would be the species that are more vulnerable than the species that are currently common.
Really interesting. I'd never thought about that. That's great. Okay. All right. Shall we move out into even deeper water then, Simon?
Perfect. Yeah.
Okay. So a couple of years ago, we had a floor talk. Some of our friends will remember. We had a face-to-face floor talk with Kiralee Moore about the voyage of the Investigator to look at Tasmanian seamounts. And Simon was also on that trip, so we thought we'd look at it again from the perspective of mollusks and seashells. So I'll hand it over to you, Simon. I'll move us on to the
Perhaps we can go back,
investigator.
perhaps we can go back to the previous
Mhm.
graph again. I know it's another graph, but the previous one.
Yep.
This was again from the Port Arthur dredging with Rob DeLittle, just showing you that the deeper you went, the deeper we went, the more samples, the more species we got per sample. So that's tantalising because we couldn't actually go any deeper than, well, in that case, 130 metres or so we got down to. So that's where I say the deeper you go, the more interesting it gets. So if we go on to the next slide, this was the old Southern Surveyor, which was the CSIRO research vessel for many, many years. And I didn't get to go on it, and I'm probably glad I didn't, because I gather it was not for the faint-hearted. They did do a whole lot of surveys for various reasons around Tasmania, including some fisheries surveys, but also some surveys of the Tasmanian seamounts where the orange roughy fishery is operating. So at the end of the fishery, when it was starting to be closed down to set up these marine protected areas, they did some surveys to find out what was living there. And then 10 years later, they went back and did a revisit to see how things were going. And then by the time I was involved, that was when the investigator had replaced the Southern Surveyor, and they went back again. So 20 years from the original survey, we went back to the Southern Seamounts. And the Investigator is a fabulous ship because it's really stable, which you need in those southern waters because it's pretty rough, can be pretty rough out there. But this undersea map shows the terrain on which the island of Tasmania sits, and you can see that we're part of the continent of Australia, but the continental shelf, Bass Strait is entirely lying on top of the continental shelf, so the sea there is seldom more than 60 metres deep. But off Tasmania, and particularly off the east coast of Tasmania, you can see the continental shelf drops away really quickly. So Cape Pillar on the Tasman Peninsula is about the closest point to the continental shelf, so it's not surprising that that was a real hotspot for finding some of these deep sea mollusks. But the CSIRO voyages went into that sort of, I forget what the area is called, between Tasmania and New South Wales is that sort of canyon system. They were doing fisheries surveys in there, going right off the shelf. And then when I was involved in the investigator, we went to the Tasmanian seamounts, which are off the southern end of the shelf, where it gets sort of lumpy and there are isolated old volcanoes basically sticking up on the sea floor.
That's an amazing graphic.
Yeah, so the sea floor there is anything from 2,000 to about 3,500 metres down, 3.5 kilometres down. And the seamounts get up to about, I think the closest would be about 600 metres below sea level, but most of them no closer to the surface than 1,000 metres, so 1 kilometre down. But there's lots of life down there, so that's the one thing that we've learned in the last 150 years since scientists have been studying the deep ocean. Prior to that, people assumed that beyond the photic zone, beyond where there was photosynthesis, they assumed there'd be no life effectively. But so when the first dredges were sent down, or first grabs, people were amazed that there were actually things living down there. And it's one of those truisms that we do know more about the surface of the moon than we do about the deep ocean. What's found in the deep ocean is just amazing. It's amazing that life has adapted to those really punishing conditions down there. So what this slide shows is some of those mollusks that came up from the Southern Surveyor voyages. And I think at the time, most of these were undescribed. Some of them are now named. They tend to lose their colour because they were sitting in ethanol or formalin for 10 or 20 years before I got to see them at the Tasmanian Museum. We were very lucky to have these samples sitting there waiting to be looked at. So I worked my way through them with help of a volunteer, and they're now all in our collections.
Very beautiful.
They are beautiful, yeah. And these are, I think, also from the Southern Surveyor voyage. That's right. Yep. And at that stage, when I did this, when I prepared this slide, about 50% of the species were undescribed. There are people working on these now. I'm not really part of that. I facilitate that through making sure that the specimens we have at the museum are loaned generally to researchers at the Australian Museum, where they've got a bit more of a program going for describing these deep sea species. And what they're particularly interested in these days is DNA. And that's where the Southern Surveyor trip was so helpful, because with these that's where the investigator trip was so helpful, because with these previous trips, the standard procedure at the time was to put everything, a lot of things anyway, at least, into formalin first before they were transferred into ethanol. It's great for preserving the flesh of the mollusks, but it actually destroys the DNA. So for the current generation of scientists working on DNA to try and work out what's related to what, having specimens, including the animals, but not being able to get DNA out of them was really tantalizing. So more recent trips like the Southern Surveyor trip, we've been able to collect things directly into ethanol, and then they're useful for DNA as well.
Mm. Amazing. I think there's a few slides here of the process of collecting.
Yes, so that's the dredge, which is a little bit better, a bit more sophisticated, shall we say, and heavy duty than the one we used off the Rambler. And it needs to be, because it's going down 3 kilometres sometimes.
Mm. Mm. I think there's some specimens there.
Yes, I think I don't actually have in this presentation. I don't have many pictures of what we collected from the investigator trip down south, but it was really it's a fascinating. It was a really great opportunity to see what was involved in collecting those samples, because you see a pot of shells like that and think, oh, well, that's quite interesting. But when you think of what was involved in collecting those mollusks and the other creatures that are being brought up, it's a huge amount of infrastructure, a huge research vessel, and all the research infrastructure is behind the vessel. So the staff that run it, the facilities that are required to just to get a sample, just to get a dredge down there and to know what you're doing. So very high technology, high tech basically.
Indeed.
Ship itself is
got a photo of the team there.
Yeah, the ship itself is potentially rolling around in 5-10 metre swells, and yet it's able to position itself to within a metre or two accuracy. If they want to go to one particular spot on the sea floor that may be 2 or 3 kilometres down, they can do it. It's just quite amazing.
Mm.
But that was me there, yeah, on the Investigator, December 2018. Yeah,
Fantastic. I imagine there's still a lot of work to do, Simon. What would be the sort of the big things yet to be done, do you think, from your perspective?
Well, at the moment, when I've tried to identify things from this voyage, for instance, I've relied on comparing what we've got to what's been found in New Zealand, what's been found off Antarctica, and what's been found off New Caledonia, because those are the areas that have been worked up probably better than most of Australia at the moment. So we're a big country, but we have we've just got the one research vessel that's able to do this, and relatively small numbers of taxonomists and scientists. So we've still got our work cut out describing what we found, because as that previous slide shows, perhaps up to half of the fauna down there, the mollusk fauna, is still undescribed. People say, well, what's the point? And I'm not one of those people that thinks that nature has to have a value, or even that scientific endeavour has to have a direct human economic value. I think doing science for science's sake, and just for the sheer wonder of knowing more about what's living on our planet and in our deep oceans, to me is justification enough. It has to be said that this wasn't a mollusk voyage. This voyage was doing far more than that. In fact, that the direct sampling of what was living down there was really something they did between other jobs. So they were basically mapping the terrain and looking at the condition of the seamounts in response to the closure of the orange roughy fisheries. So the voyage itself had a very applied motivation. The mollusk stuff was a bit of a sideline, but for me it's something that's well worth doing, just for the sake of science and for understanding our natural world.
Indeed. Thank you very much, Simon. I guess our friends would want me to ask you what happened to your childhood shell collection before we finish up today. So did it come to Tasmania with you, and do you still have it?
It did. It followed me to Tasmania, yes. I brought it over in a shipping container when one was coming this way. And there's a little story there, because I documented everything in my collection, and I gave that to the customs people, and they looked at it. Obviously it didn't mean the names didn't mean anything to anyone other than one name that came up, which was Tridacna, which is the giant clam. And when I got my specimens, it wasn't a listed species, but these days it's listed on CITES, so you're not actually meant to trade or traffic in it across international borders. And so they said, right, we're going to have to seize your collection. So they seized my collection. It's not that I wasn't allowed to bring it in, but they had to go through the process. And then they said, right, okay, now I need to find someone who can who can identify this shell and check that it's a species that you're allowed to bring in. Who can we find who could identify the shell? I don't know, we'll ask Dr. Simon Grove.
That's a fantastic story.
I was both the person that brought the shell in and also the guy who said, yes, that's what it is. But yes, my Tasmanian seashell collection is, my childhood seashell collection is with me in Tasmania. My Tasmanian seashell collection, I've now donated to the Tasmanian Museum. So that's now over in the collections at Rosny, and parts of it are on display in the Earth and Life Gallery, because we've got a little cabinet there which features every species that you could expect to find on the seashores of Tasmania.
That's great. So if our members want to find out more, they can go into TMAG and have a look there. And there's also your book and a website or an online catalogue, I suppose you'd call it, isn't it?
Yeah, so the book is really limited to the species you'd expect to find on the seashore, and it's pretty much a one-to-one. What's in that book is also in the cabinet in the museum. The website lists a whole load more, but it lists a lot of those deep sea species as well, and I've got photos where I've managed to get hold of them. So that's about 1900 species as opposed to about 350 or 400. But yes, plenty of information out there these days.
That's fantastic. And I have to say, I was lucky enough to be given a copy of Simon's book a year or so ago, and I use it all the time. It's a fantastic resource. So available from the TMAG shop, I think. So we'll do a plug for that.
Thank you, Janine.
Okay. Well, look, thank you so much, Simon, for talking to us for this period of time. It's been wonderful. So I think the only probably less than smooth part of the recording now will be as I hunt for the stop button on the recording. So thank you very much for joining us, and I'll see if I can finish it up now. And yes, we hope that many of our friends will get to see it. So thank you, Simon.
More to the point, I hope that many of the friends get to go out on the beach and enjoy Tasmania's nature.
Thank you. Okay.